Okay. So we have here the famous pipettes. So what I'm gonna do is put some amount here. It doesn't matter the amount. Just something that I feel comfortable with.
¿Más alta?
Hmm?
No, ya tenía la.
And then you take the liver. It's the liver, no? So what I'm gonna do is I'm gonna peel. I'm gonna probably use this. I'm gonna peel all the white.
Let it dry because it's in ethanol. Right
Yeah.
now.
All the white tissue. You see the white tissue here, right? That white tissue is very bad, because it has a lot of fibroblasts, and these fibroblasts grow as crazy. So then, no, you need to remove that. Well, these ones are very small, but
Those are scissors. It's
Yeah.
okay. It's okay. Go in.
is it
Ah, you
so?
wanna- Yeah, go in.
So what I'm gonna do, these are scissors, right? So I'm gonna try to remove that white stuff. I don't want that thing. And you can do it with the scissor, or you can do it with the other twister like this, for example. So you hold the organ, and then you do it. But the bottom line is you don't want that white thing. I think that now it's clean. So then I'm gonna move it to here, and now there I'm gonna extract the cells. Let me see if I can. Yeah. Now I'm gonna use the syringe. Normally I use an small syringe, but this one is okay. And the needle. So then what I do, so I use the tweezers to hold the organ like this, you see? And then I break down the organ in multiple places like this, in a way that now when I flush it, it's gonna have multiple holes for the medium to get out. It doesn't matter that there is air here, but I can clean it a little bit. So and now you are comfort. Let me see. There is something here that is floating that I don't know. Because the more you remove the white stuff, the better. So then you just pinch or whatever it's called. Yeah, this syringe is too big, but we wanna do it. So you just pinch it and then flush it, and you're gonna see that the cells the spleen has to be almost white, almost transparent. after doing this. You see the cells? So then you do it in multiple places. By the way, the spleen is okay, but it's not very big.
No, not as funny.
You try not to break the organ, because then it's more difficult to extract the cells. So in that way you try to get the majority of the cells. So for example, here I want to target this one. I'm gonna go like this. You see? But I think that is okay by now. I mean, now it's okay. Let me do something here. No, it's okay. See the see now this. Let's see how I can. You see that now is almost white. So then that's okay. Now I'm gonna use another tube to grab all this. So the tube are here. Can I have a new one? And now we need to count it. Now counting this is not trivial, because what happens is that these cells, so there is a lot of red blood cell there. So then counting them, in order to count them, you need to differentiate the one that are red blood cells from the one that are splenocytes. It's very difficult. So then the red blood cells tend to be like yellow when you stain it with the trypan blue. But to be honest, I never feel like comfortable with the number. Now the number has to be close to 100 million. So I'm gonna leave everything here, because I don't think so I can retrieve a lot more cells from the spleen. And you see the spleen? You see that it's kind of, it's not red anymore, right? So there is some areas that are red, but the majority is pale. So if I'm gonna do something else, is try to get more cells from this end. But I don't think so. I don't think so we need it. I think that we're gonna have enough cells here. So then let's count it. Let's put everything in the back. or I tell you something about the pellet. Something that I learned, and maybe it's important that they learn too. But let me do this one. The medium has to go. And then after that, we have to dislodge or whatever is called the pellet. So by removing the medium is really critical. Because if you left medium, now the peg will be diluted. So now the pellet. So the pellet has to be dislodged. Like
Easy.
this. And you have to do it really well. Sometimes what I do is like this. Sometimes I do like this. But the pellet has to be, you see now it's better, but it's still. So the pellet has to be this low. You cannot do too hard because then you're gonna get pellets everywhere. So you have to spend time on this. So the way I think is ready if I can like move the, you see that it's moving. So now it looks like the pellet have been this way. Now, so now we have the pellet disrupted. Now we're gonna do 500 of the peg, and we're gonna do this one working, and we're gonna do. I'm putting it slow.
El de arriba es para succionar y el de abajo.
Okay. Then, so I'm gonna put it in slow. So I'm gonna do the peg first, 1 minute.
1 minute.
And then right after that, 5 ml
5 ml
of
3
medium
minutes.
in 3 minutes. And then after that, 15
15 ml and 1
in
minute.
1.
So the whole thing is just half.
We dilute it, so I'm gonna just mix it. We mix it,
Mm-hmm.
but just in case. Now, Juan, you
Yes,
have
I have
them?
the tiger right
Yeah,
now.
let me know when, like a 15.
Yes.
Yeah. This part is the mess because this part is the one that you cannot control very well, you know. Because, for example, let's do a practice here really quick. For example, the drop has to be something like that, you see. But sometimes you go more than that, you see. But something like that has to be drop. So then the trick that I use is this. So instead of doing like that, I go like this. You see? So, but the thing is, you see? So I am not pushing the piston. I am just doing this.
Mm-hmm.
You see? So I end maybe pushing the piston, but I try to do it that way. So 500.
Yes.
And you do that in the vortex.
Tell me when I start.
Yeah. So, for example, like this. Now, what I was saying before, so it depends where you hold the tube. If you hold the tube here, it moves less. You see? If you hold the tube here, it moves better. You see?
Mhm.
If you hold it very tight, it doesn't move. So you have to hold it. You don't want the tube to fall down, but at the same time, you want the tube to move, right? So let me get into position. Now, if you want, we can.
Start right now?
Yeah.
Ready.
Just let me know the. Huh?
Quedan 45 segundos. ¿30 segundos? Sorry.
Okay.
20 segundos. 10 segundos. 5 segundos. Ya está.
Now,
Ahora van a ser 5.
now I'm gonna do the 3 minutes, right?
Ajá.
And it's 5 ml, no?
Sí. Yes, correct. Now, here we go.
Ajá.
Ajá, ready.
Oh, come on.
Quedan 2 minutos con 30. Dos minutos con 10 quedan. Ahorita momento. Vamos,
Manuel.
quedan, sí, 2 minutos. Apenas pasó 1 minuto.
Oh, apenas pasó un
Ajá.
minuto. Ah, sí. Sí, quizás le di mucho, pero debió haber ahora aquí quedado como menos de un minuto. ¿Cuánto queda?
1 minuto 40.
Oh, sí. Lo diluí muy rápido.
Queda 1 minuto con 30. Ahora se queda 1 minuto. Segundos.
Y ya lo terminé. Y ahora viene lo de 15. ¿no?
Ajá, 1 minuto.
¿Ya?
Ajá.
Este sí voy a hacerlo más
Sí, un poquito
rápido.
más
todavía.
rápido.
Sí.
Ujú. Segundos.
Este tengo que darle 5 más.
20 segundos. Segundos, 5 segundos y listo.
O sea, ya este era el último que teníamos.
Sí,
que-
hasta ahí ya centrifugamos.
So the last 3 ml in 3 minutes.
5 in 3 minutes.
5 in 3 and 15 in 1.